Okay. Well, thank you, David. I guess I'll start by sharing my talk. This should be without problem. Let's see. Is this working? I need some confirmation. You're seeing my slides. Okay, perfect.
Thank you.
Okay, thanks a lot to the organizers for providing me this opportunity to share basically what has been the results of decades of work, not of one person, that indeed from a team of researchers that we have to say have been an example to the field of complex traits in the sense that we've working together already for more than a decade and showing results. Also, I want to take the opportunity to congratulate you for putting together this excellent program during these 3 days. We'll look forward to follow. And I think integrates very well with what we want to do. And that's the focus of my talk. That's basically how we can translate genomic discoveries to the clinic with emphasis on the musculoskeletal field. So, let me see. I don't have control. Okay, yeah, if I click, it goes fine. Are you seeing my mouse?
We do see your mouse.
Okay.
Yeah, your arrow.
Now I think this one's better. Okay, so the outline of my talk, as David already suggested, and once more, thanks for your very kind presentation, is to sort of give you an idea how we came to be with our genomic discoveries. I want to emphasize on the complexity of biological systems with emphasis on the musculoskeletal system, which was also very nicely introduced by Milana. That's basically the reason we have to have these new methods that need to be implemented in order to deal with this challenge. I want to focus on showing how this cannot be done by one group of experts. Actually, it has to be a team of multidisciplinary team of experts. And this requires to bring different techniques and analytical approaches to do this. And I want to end providing some translational examples. I will be probably disappointing you in the sense that I won't go in deep with any of them. But I think we have the opportunity to see some of these along the talks during these 3 days. But also, please feel free to contact me directly or any of the people I will mention to get additional information on those examples. So this is just to be provocative, to follow on what the field is working on. So where are we and where we are moving? Again, this is about translational genomics because it's about bringing DNA research into the hospital and most important in staying there. That's the pathway to genomic medicine. And I always like to start with this very nice illustration from Eric Green published in Nature already some years ago, showing the enormous challenges that we're confronted to do this. And it moves from understanding the structure of the genome, which was done by the Human Genome Project, how we understand genome biology, and how we understand the biology of diseases. And this is a crucial step that will allow us to advance medical science and actually to improve the effectiveness of healthcare ultimately. Now we see that, well, probably we are not yet there because it's already 2020 and we are here. And we want to get here. Why does this not materialize yet? And that's basically because we have some bottlenecks that we have to deal with. And it all builds up from what happened in the last decade with GWAS, and we've come with a long way, because if you look back in 2005, this is what we knew about the genetics of complex traits. And you can see already how this genome looks like now, powered by associations telling us that there are links with disease. So GWAS has revolutionized the approach. In very simple terms, for those who are not familiar with GWAS, it begins all with the throughput that the new innovation in technology brought with the microarrays, where you can genotype now millions of SNPs of one individual simultaneously. And when you do it within a well-defined study in your samples of interest, you can test for associations. And many of us who started by ourselves realize we don't get anywhere just by ourselves. That already points to the importance of the complexity of the system. We actually need to bring all data together, and that we usually do with meta-analysis to get these very robust associations. We have been lucky to be centered this with our human genotyping facility in Rotterdam. And we invite you now to make use of this technology because it's really affordable. And we can really genotype for about 30 euros per sample, what at the beginning was more than 600 euros. So again, I want to show you the opportunities that genotyping provides and what can be used for these translational efforts. So I'll mention briefly the GFOS consortium, the Genetics Factor of Osteoporosis Consortium, already started in 2007, ended in 2012, and that's why I say with pride, we are very successful, because close to 8 years later, we are a very big family still working closely on this. So indeed, the path to success is really embracing collaboration. Now, this is just to show the findings that we have on bone mineral density. And you see that there are multiple efforts, not all of them, of course, depicted here. But what we can show is that every time we join forces, and that was within the GFOS consortium, we see how we have this very big leap in discoveries. So collaboration is key, working together, that's what brings us forward. Now, to emphasize, why do we need these large GWAS? It's not just about getting these very marvelous significance, low p-values. It's really about the complexity of the underlying biological systems. And together with Otima Kitty, we came with this graph trying to explain how an individual comes to the event of interest, the clinical outcome, that's fracture. And you will see that there's no real one path taking you to fracture. Actually, it's a complex melting pot of biological process, all of them which are subject to genetic influences and also environmental influences. In addition, it occurs within a system, and the system can adapt. So what's going wrong in one side can actually be compensated by another. This is a contrast with what happens with monogenetic diseases, where, for example, you have a very big alteration on material properties, like, for example, defects on type 1 collagen, which can have such a large effect that there's no possibility to have system adaptation, and just this mutation will lead you to fracture. In contrast with the complex forms, you have that we have different interactions also with the environment that makes this very complex and heterogeneous. And in addition, it's not just about the musculoskeletal system, but what's happening outside. And you have to take into account potential pleiotropic relationships, and also the limitations that are brought up by confounding. So all this heterogeneity and confounding means that actually size matters, and to overcome this, collaboration is key. Luckily, we have game changers in the field, And one example is the UK Biobank resource, but there are many more like these evolving around the world, where we have sample sizes that are bigger than our previous consortium altogether. And you see here what old Manhattan plot looks like now. It's basically a grass plot, very not informative anymore, but telling us that there are many findings that are occurring. And what have we learned about these findings? Well, let us think that when we first moved from the common GWAS to the rare variants, we did find them, but well, the effect sizes were bigger on average, about 4 times bigger from what we found. But as we increased sample size, we indeed started to discover more of them. But you see that the novel ones are always appearing with effect sizes that are smaller. And this is even amplified when we have 500,000 samples. You see that the novel ones in here in blue are always having effect sizes that are smaller. So yes, more discoveries, more biology, but with effect sizes. And the question is, what do we know about the new biology behind them? And that's basically very little. So we have to take new approaches to take this to the clinic. And that means several types of integration from data from different origins. Not anymore just doing genetic epidemiology, but basically bringing functional assessments at multiple omics levels, all of them integrated at the cell, animal, and human models. That can be then helping us to identify what are the actual genes that are underlying the signals to be used for translational purposes. Now, this does not mean that we have just done genome-wide association studies and nothing else from a perspective of just genetics. No, we have already partnered with functional experts that have already provided insight, and we have a very large set of examples of how this can be done successfully. For example, our collaborations between the GFOS consortium and the Origins of Cartilage and Bone Disease Mouse Phenotyping Consortium. The downside from this approach is that we are doing it one by one. And despite that they already have the infrastructure to actually phenotype hundreds of animal models at the time, we need to bring all these data together in a systematic way and not just illustrating on every publication one example at a time. So the big question is, what's next? Continue increasing sample size? Yes, that's something that we need to do because there are traits that need to provide us more information. BMD only tell us about involvement in bone biology, but does not tell us how. So additional GWAS on more specific traits are needed. We always have the problem of limitations of sample size with that strategy, but this is also an opportunity to take the BMD loci that have been identified in a new candidate gene approach and explore these in new phenotypes that will provide more insight into biology. But just increasing sample size is not the core plan. We have challenges ahead because we don't get a lot of answers just by doing that approach. So we need to embrace multidisciplinary collaboration. And that's what Jeff's Gemstone, the Genomics of Musculoskeletal Traits Translation and Network, and other efforts that are also starting to be set up in this line. It's a cost action and infrastructure grant where basically being overwhelmed with the very large number of genetic discoveries coming mostly from the existing GWAS, but now the mega GWAS like the UK Biobank, but also from whole genome sequencing on monogenetic families. The time is ripe to get all these discoveries and bring them one step forward. And that requires the incorporation of bioinformatic approaches, the use of cell and tissue models at the different omic levels, but also the integration from the animal organisms. And that's why this workshop is providing you this whole view of the potential where we can go to take these discoveries into clinical translation. Very important, we also have to learn about the phenotypes, how they relate from all these out, what it's relevant when we apply the different models to actual the human processes. And again, we are confronted with the complexity of the relationships, but also the very big data that's there. And that's why we need novel analytical methods to do this. Very succinctly, we have 6 working groups. We invite you, all of you, to participate if you're already not making part of Gemstone. There are evolving papers providing insight in what each of these working groups is trying to achieve. And I have to highlight the one on functional investigations, because this one is creating a roadmap of the loci that we want to study in a coordinated way. The monogenetic conditions is telling us how we can get insight into the complex forms from the monogenetic forms and vice versa. And the phenotyping is crucial because it's telling us how we can do this integration, basically what discoveries are relevant for the human translation. And of course, the analytical part from the bioinformatics, building all upon the inventarization of data that's out there, and trying to reach basically those settings that will allow us to do translation, like reaching out to stakeholders that can be, for example, the pharmaceutical industry, but also patients. With the essence of this workshop, we have one big question. Can we afford not modeling the complex of underlying these biologic and disease processes? It's complex. We cannot just think we can go from genome to phenotype in a simple way. Actually, we have to take all these different interactions. And that's why we will look forward to receive insight on how new methodological approaches can allow us to model this data. And you will have this slide from Henadis Ross-Chupkin showing us how this can be integrated, just to give you an idea on how we can take all these omic assessments and make a meaningful outcome from them. And shortly also, I want to emphasize what we have managed to bring together within the big data working group, mostly led by Douglas Kiel in Harvard University in Boston, which brought us together with an existing project in the Broad Institute that was the Musculoskeletal Knowledge Portal that was built up initially for diabetes and now is evolving to other traits. So we could create the Musculoskeletal Disease Knowledge Portal. And this is a tool that's already available for you to be able to mine the genomic discoveries initially, but also have an interface that allows you to integrate also the findings from other omic assessments, not only in humans, but at the different levels. Have to emphasize that this is evolving. And for example, this is the new setup that you have. And there are already, we've presented before workshops on how to use it. And these are to be reached also through the IFMRS website. And I encourage you to do so. Now, during the coming minutes, I would like to just emphasize some translation examples. This is really a selection of what's going on. Just wanting to be provocative, and I want to finish early so we can have a time to brainstorm around these possibilities and think together on how to move forward. When we think about translational genomics and moving to genomic medicine, You have several potentials that you can have, and this is nicely illustrated by this paper from L.S. Gini, recently published, where there are multiple approaches that you can follow. I'm going to do this presentation from, of course, the bone, but also providing some examples of what we have learned from other fields and the potential for other outcomes. So when we think about target discovery, we always go back and think about the limitations that are proper from the pharmaceutical industry, and how it is a very, very long process, a high risk process with a very high proportion of selected molecules that are deemed to failure. And this paper, this is really a seminal paper from Matthew Nelson from GSK, showing how if you provide the support of human genetic evidence for the choice of targets, you will double the success rate in clinical development. So actually taking the information of genetics will increase your likelihood of success and avoid failure. And this occurs at different phases. And that tells you how these, if you incorporate information from the GWAS database and from OMIM to prioritize molecules. And we were very happy to see that one of those was taken from one of our discoveries, the bone mineral density GWAS, how this can evolve into a treatment and how using that information actually really takes you into a path of greater success in the selection of drug compounds. Now, of course, when you think about genetics, you think about risk prediction. This is usually done by genetic variants that are clustered together and sum up in the forms of polygenic risk scores. You see that the polygenic risk scores usually follow a normal distribution within the population, where the average number of, the mean number of variants are going to be clustered around the mean of the population. So a lot of individuals are going to have just an average. But you will also see that there are sectors of the population that are going to have, if you're talking about risk, very few subjects that have a low number of protective variants. And on the other side of the spectrum, a very few numbers that have a very high number. And if you look at the gradient of risks between the extremes, you see it's going to be very, very, very high. So first of all, we have the possibility to discriminate between low and high risk. That's something that we cannot do with the use of independent variants because we know they have very, very small effect. But when we bring them together, they are actually becoming informative. This is not new. This is something that we were already doing in 2012, just to illustrate in the context of lumbar spine BMD, how those having the mean number, the mean BMD are really confined to having a mean number of BMD decreasing alleles, and how you have these very few number of individuals that will have very high BMD and very low BMD. And this correlates also with the clinical outcome, that's fracture. But then again, we have been continuously thinking that the predictive value is not very big when you put it in the context of the existing clinical scores. But I need to say, we're starting to show that's not the case. I'll provide some examples of that in the sense that they are indeed providing information, and particularly in other diseases where this is really demonstrated. Sticking to the BMD field, this is one of the first efforts trying to predict osteoporosis and fracture using a whole set of studies. You can predict how well the eBMD, this is BMD measured by estimated or calculated by ultrasound from the heel. It's what we call the poor man assessment. But it predicts very well the heel BMD of the individuals that's measured. And also, it can predict those individuals that are at risk of osteoporosis. So if they have low BMD, estimated BMD predicted by genetics, you see that they also have a high risk of osteoporosis and also for fracture, showing that it works. Now, we have different risk scores, genetic risk scores that we have. And what we would like to see is that there's consistency between them. And that's what this paper has shown. When you apply it for the prediction of fracture, those that have been estimated from heel ultrasound versus those that have been estimated from DEXA. And this is very recent work from the group of Brent Richards, showing very nicely how these can be taken into a step forward into the clinic. And of course, you want to build up from the proof of principle that the genetic risk score, what's called GSOS, and which has already been agreed, it's going to be shared for the community, that you can use these GSOS for your research purposes. It predicts very well, has a good correlation with the measured heel ultrasound as a proof of principle. But what's very nice to show is that if you compare screening strategies for osteoporosis without using GSOS and then without using and with using the genetic heel ultrasound information, you see that the sensitivity and the specificity is kept. Actually, you have some gain in specificity, but probably the biggest value is about reassurance, in the sense that here you have the individuals that require additional steps. So for example, a measurement of DEXA BMD that we know it employs resources is always lower for the genetic source, meaning that we are able to reassure the low risk of individuals. And I think this is an important lesson. We don't have to focus only on individuals of high risk. Actually, BMD does that very well, but we have to think about those individuals that are reassured as another gain. Now, this was about prognosis. So I want to go back in time because this paper was actually published in 2018 and tell you about an approach that can be used at different levels. The example I'm going to show you, it's at a clinical level. That's the use of Mendelian randomization to examine the clinical determinants of fracture. But I also want to emphasize that this Mendelian randomization can be used also at the assessment of functional aspects. So you can do this, for example, applying it for gene expression. And the essence of the Mendelian randomization is that it allows you to tackle the problems of confounding that's intrinsic to any design of a study. And also, it allows you to infer potential causal relationships, which is critical, not to predict, but to understand biology. So we use polygenic risk scores to improve the research in the clinical arenas, address this problem of confounding, and identify effective interventions in the clinic, because once you demonstrate that there's a causal relationship with a risk factor, if you intervene that risk factor, you're going to have a clinical effect it's going to be successful. So we took basically the assessments that we know from clinical risk fracture, and we looked first at those polygenic risk scores that are related to fracture. And then we did it for all the other, using summary level data of all the other risk factors. We could look at the genetic correlation between the factors. That's basically the shared heritability, and see how are they related in genetic terms. And we were surprised to find that the well, not surprised in the sense that we did expect that BMD was going to be highly correlated, sharing genes that are also involved with fracture, but that the other ones that we had shared very little genetic heritability, genetic correlation or shared heritability between the factors and fracture. And what we showed also is that the relationship between BMD and fracture is causal. And probably if you want to have a successful intervention, targeting the lower of BMD is going to be critical. And while the other risk factors may be informative, they are not really causal, except for decreased grip strength, which subsequent studies, because here it did not do correction for multiple testing, but more recent work has really replicated this and showing that it's significantly related in a Mendelian randomization context to fracture, showing basically that BMD and the muscle assessment are really causally related with fracture. So we conclude that BMD is a pivotal cause of osteoporotic fracture. And among all the studied clinical risk factors, the interventions aimed at increasing BMD are likely to be successful to reduce fracture risk. And our work had quite some attention because among this list, you also had decreased vitamin D levels, actually, where we could show that lower vitamin D levels and estimated calcium intake from daily sources were not associated with fracture risk, which this is a message that you can take. This is evidence against indiscriminate vitamin D and calcium supplementation for prevention of fractures. So what else can we do? And we can exploit the use of polygenic risk scores. We can do that for response to treatment, for stratification. And I would like to take one step back from the musculoskeletal field and then look at diabetes, because they have walked a long way in this direction. And what they have achieved basically is seeing that diabetes, as it's known clinically, let us take type 2 diabetes, it's actually a melting pot of different types of diabetes, which will take different and distinct types of pathways. So without doubt, genetics can guide the interpretation and understanding of the underlying biology, which in itself is the same that it's approached for the use of medication, for example. So treatments are basically targeting approaches. So knowing the biology will allow us to see if a specific intervention or treatment is going to act better or worse in a pathway. And this is what has been proposed as the palette model, where basically you have different diabetes and component pathways, and you're going to see different groups of individuals that have a greater characterization. So you take, for example, 4 individuals, you will have that one that's very clear, because actually this type of diabetes, of pathway, is going to be very prominent in this individual, while not the other pathways. And you can cluster them very good. But there are going to be others that are going to have different levels of pathways and contributions that will be very difficult to assign to a specific group or groups of individuals. And then you can have individuals that currently don't have the disease, they don't have the pathway, but in time actually can move into one of these palettes based on having the medical condition, in this case diabetes, or not. So indeed, this is something that we would like to do here in a nutshell. This is what we do in clinical practice and how we can apply a polygenic risk scores in that part. This is the graph I showed you already. This is basically a gradient of risk of individuals. And what we did was take each individual in one column, and then you see that, for example, this individual has mostly green variants. That's low risk. So he corresponds to having a low polygenic risk scores and having low risk for osteoporosis or fracture. While individuals on this side are going to be mostly red, if you see, because they have an enormous amount of variance. And then you see individuals that have an average between green and red. So this is true data. This is actually how it looks. But what we are looking for is for clustering into WNTs, for example, the WNT pathway, because that will determine a specific response to treatment for those outcome or treatments that are related to, for example, the WNT pathway. But then again, I emphasize this is hypothetical. That's something that we're looking for. So what we would like to do is have components of osteoporosis in pathways. We would need a score for diagnosis. We could still be working with BMD, and then, of course, 6 individuals that fracture without having a low BMD. So this could be working, but we want to find these individuals that cluster separately in that context. And that's what diabetes allows us to do. And there's a project I will acknowledge towards the end. That's the Fidelio project, basically looking at the etiology of the diabetic bone. We look if the relationship between BMD and fracture, which is paradoxical. Diabetic individuals have higher BMD than the average of the population. But despite that, they have higher fracture risk. And we want to know if it's causal. And if you do this just by serum glucose, you're going to see how confounded the relationship can be. But when you take it to the genotype, the base sampling, you can see that the relationship is still there. It's not there anymore. So the BMD is not there. Now, this can take even better when you have precious phenotypes that cannot be used. And for example, in osteoarthritis, and I want to take one example that's in how Professor Karasik mentioned about the Generation R study, which is a pediatric cohort. And we have very nice measurements that we have determined that could be related to osteoarthritis. And one would like to know if these pediatric relationships, either with MRI or with other aspects, are related to the risk of OA. And we know that the clinical hip OA is not applicable in children, but we know that there are children with a high or a higher value of this level. But what we can do is take the genetically predicted hip OA and see if on the extremes of the distribution, which is called recall by genotype, you see an association with this relationship with epidemiological relationship with osteoarthritis, and we see that it's not causal. So it could be related to confounding. Now, to end, this is just to provide an essence of the recall by genotype, how it works very well because it's analogous to Mendelian randomization. And below every single polygenic risk score, you are going to have that the confounded associations you see with the measured level are actually going to be there. So basically, we're tackling the issue of confounding while we are still keeping the relationship with the outcome. And we can focus in these individuals for distinct purposes. So what can we do? We have this relationship we already established, but we can take an overlay, and that's what we're thinking. So here you have the individuals that have osteoporosis. You see all of them have osteoporosis, but then you see what their PRS is. And then what we've seen, and there's work showing that when you have a polygenic risk score that it's not high, but you still have a very severe phenotype, that is a very good indication to look for a monogenic mutation. And these are individuals that you can choose. But you can also use molecular phenotyping. So like in this example, look if the cases that you have or group of cases are related to cholesterol measured genetically. And we see here that basically you don't see it. While on this one, you see a cluster of individuals that, for example, this is hypothetical, be related to inflammation. And then you can think about osteoporosis that it's related to inflammation on these type of individuals. So that's another way we can expand for it. And to finalize, well, looking into the future, you can see that this can be used, this strategy of recall by genotype, to perform deep phenotyping. Because if you are going to start a study, you're always going to have the problems of confounding that are going to be affected. While these, you can do dynamic sampling with a very small investment that can be just using the genotyping and questionnaires for the initial phase. And that's all you need, GSA genotyping and the questionnaire, and that's all you could need. You can do it for very big numbers and then prioritize the very expensive or elaborate assessments on very specific group of individuals. Okay, so to finalize, we're working on creating a coordinated roadmap of integrated functional assessments that will translate in genomic discoveries. We know that there's biologic clustering of these identified loci, and they are revealing new bone biology and many translational opportunities. They feedback each other because you can have optimized polygenic rescores once you know more about the biology. These biological pathways, we hope they can lead to new molecular definitions of bone disease. We can use this genetic evidence to accelerate the drug discovery process. And the way forward is really to study the mechanisms at the cell, tissue, organism, and population level to bridge the gap between the concept and translating to the clinic. So thanks a lot. I think I'm now over time, so I'll just go through the slides. Acknowledgement, a very big number of peoples and projects and
You good on
consortia.
time. You have 5 minutes, Fernando.
Okay, but also for the discussion.
Especially since here you discussion will wait. It will be in 40 minutes or so.
Okay, perfect. So yeah, well, I can acknowledge the group, our genetic laboratory in Erasmus MC, the Fidelio project studying, it's an ITN studying the diabetic bone from multiple perspectives, and at least 14 research centers throughout Europe. I'd like to emphasize Sankatan and Rolin Lee, who are working on this project. They are our PhD students. Then moving to Gemstone, well, the people involved in the management and the coordination, working group 1, which I said is critical because it's where we are all sitting on trying to identify the resources, both of expertise and data sets. They are Nerea Alonso and Ivan Soldatovic, and Ines Fossi and Barbara Overmeyer, who are coordinating the working group 2 that's based on phenotyping. We have a beautiful paper on musculoskeletal phenotypes and how they are can be extrapolated from the cell level all the way to the human population level, which I invite you to follow. We hope it will be published during the first trimester of next year. Working group 3, which is about monogenetic conditions, led by Oti Makiti and Melissa Formosa, which we basically envision trying to go back and forth between the complex and the monogenic forms. Also producing their roadmap paper for the working group. No need to introduce Professor Karasik, who together with Martino Lohner are leading the functional investigations working group. And this also has a paper that's close to publication, making the first steps to this roadmap. And the bioinformatics working group, which is led by Milana and Michael, who are really trying to bring, and I think it's also the essence of the workshop, the new analytical approaches that are needed to take this forward. And at the later phase, we are going to work together with Klaas and Anna-Marie to take the translational outreach of the consortium to the different stakeholders. And of course, the people that keep it moving at all their levels, the science communication officers, Patricia Cachayar and Ines Alves, our project manager, Fiorda Koromani, Ken Soy, who will also be talking here at the meeting, who is our STSM coordinator, allowing us to share expertise between labs. And I can tell you already, if he's not mentioned it, I'll ask him to mention it again. We have the opportunity to take young researchers and move them around the lab, and also looking at the different possibilities for the exchange of young researchers and formation of young researchers with Ana Maria Tetti, and also with our Milana, sorry. And now I know I'm taking too much time with all, but I want to finalize thanking my vice chair, Evie, who has helped us shape up this. So again, we are looking for functional work to be published on the JBMR. We invite you for this call, and I'm also open to questions. So